Dear students, Assalamu alaikum to all of you. Our today's topic is secondary growth in flowering plants. Secondary growth in plants. It means that some specialized cells are present in a plant which are responsible for secondary growth. Specialized cells. These are responsible for secondary growth. And this secondary growth, we call it as lateral meristem. Lateral meristem. Lateral meristem. Now, when there is secondary growth, it means that there is cambium. When there is secondary growth, it means that there is cambium. So cambium and core cambium are two types of lateral meristem. Lateral meristem types. One is cambium, vascular bundles, and cambium. Vascular bundles. First one is cambium and another is core cambium. And in case of vascular bundles, first one is cambium and another type is core cambium. So these two are the types of lateral meristem. Core cambium. Now we know that separate plant pots have separate type of growth. When we talk of growth, we talk of vascular bundles. When there are vascular bundles, it means that there is cambium. And when we talk of normal growth, there is core cambium. When we talk of vascular bundles, cambium, means that vascular bundles are there. And when we talk of cambium, we talk of vascular bundles. And when we talk of normal growth, there is core cambium. In case of normal growth, there is core cambium. Now, types of lateral meristem is cambium and core cambium. At first, we will talk about vascular cambium. Vascular cambium. First of all, we will see what is vascular cambium. Vascular cambium. And I have told you that in case of stem, this vascular cambium has, for example, it is vascular bundle and this is xylem and this is phloem. We know that there is conjoint arrangement. Conjoint arrangement of vascular bundles. Conjoint arrangement means when xylem is towards pith. Xylem is towards pith or center. And phloem is towards periphery. So this arrangement is called conjoint arrangement. So we know that there is present cambium. So the cambium which is present between xylem and phloem. Cambium present between xylem and phloem. It is called as intrafascicular cambium. Intrafascicular fascicular cambium. When secondary growth takes place, this cambium present between xylem and phloem is called as intrafascicular cambium. And phloem, because it is present in the same bundle. Xylem and phloem are present in the same bundle. So the cambium between the vascular bundles is called the cambium having only one vascular bundle. When there is a cambium, there is xylem and phloem. In between there is cambium. It is intrafascicular cambium. But when there are two vascular bundles like this, and there is cambium, the cambium which is present in between, it is called as interfascicular cambium. So we can say when there are two bundles, the cambium which is present between two bundles, it is called as interfascicular cambium. So the cambium between two vascular bundles is called interfascicular cambium. And there is also a medullary ring. There is also a medullary ring. When secondary growth takes place, this medullary ring formation takes place also. But this ring is of parenchyma cells. Medullary ring formation takes place, but of parenchyma cells. Parenchyma cells. These parenchyma cells become meristematic and start dividing further. So this ring-like structure is formed, but in case of dicots, because secondary growth, we talk of dicots because secondary growth is the characteristic feature of dicots. So when we talk about the activities of cambium ring, how does this ring act? When secondary growth takes place in dicots, dicot plants, xylem and phloem undergo differentiation and produce secondary xylem and secondary phloem. As soon as the activity of cambium begins, the cambium towards peak is more active than the rate of division. The rate of division is so fast that it crushes the phloem. The primary and secondary xylem remains intact, but phloem growth stops. For example, we will make here the vascular bundles. For example, these are vascular bundles, and this is xylem arrangement of xylem and phloem. And I have told you xylem is towards pith, xylem is towards pith, xylem, and xylem, and this is xylem. It is phloem, it is also phloem. And this makes a ring-like structure. Ring-like structure. So as soon as activity of cambium begins, the cambium towards the pith is more active. Then their rate of division is so fast that it crushes the phloem. So primary and secondary xylem remains intact and phloem is crushed. Secondary xylem is formed rapidly. So this medullary ray is formed. Medullary ray is formed. This medullary ray is of parenchyma cells. Parenchyma cells. But phloem parenchyma is absent in monocot stem but present in dicot stem. As soon as formation of secondary xylem and secondary phloem occurs, a medullary ray is formed, but it is formed with the help of parenchyma cells. So the medullary ray forms a ring which we called as, which later on appears as medullary ring. Medullary ray appears later on, appears as medullary ring. So we will see how formation of wood, and now we will see how formation of wood takes place. The wood formation is because of cambial activity. How formation of wood takes place. Formation of how formation of wood takes place. Now we know that because formation of wood takes place by because of cambial activity. Because of cambial activity. We know separate wood is formed by the presence of cambium ring. So on the basis of seasons, wood formation is of two types. This wood formation it is of two types on the basis of season. On the basis of season. First one is called as early wood. It is called a early wood, which we can say it is spring wood. It means that the wood which is formed in the early stage, it is early wood. It can also, we can write also spring wood. Now there is late wood. Late wood. And this late wood is also called autumn wood. Autumn wood. So we know that on the basis of season, on the basis of season, wood formation is of two types. So early wood, which is also called spring wood, late wood, which we call as autumn wood. So we will write this cambium is active in early wood. In favorable conditions, in case of early wood, cambium is more active. Cambium is more active. As for autumn wood is concerned, there is cambium is less active. Then there are more xylem elements. More xylem elements. So there are, in case of late wood, there are less xylem elements. Xylem elements. Then their vessels are wider. In case of early wood, vessels are wider, but in case of late wood, vessels are narrower. Vessels are narrow. Now there are dark appearances as per season. On the wood of surface of wood, there are dark appearances. I will show here the diagram. For example, like this. One ring is dark, one ring is lighter. It is lighter ring. One ring is lighter. This is dark. This is again lighter, and this is dark. So one ring is one ring is formed per year. So as for these, and these are called annual rings. Annual rings. So we can determine the age. We can count the annual rings, and we can on the basis of annual rings. So we can write on the basis of annual rings. Annual rings. We can determine the age of the tree. The age of the tree. Because one ring is formed per year. So we know that density of wood appears like in dark bands. Because one ring is formed in 1 year. So low density and is lighter. In case of early wood, there is low density and it is lighter in color. For example, it is late wood, it is early wood, lighter color, and it is dark wood, dark color but late wood, dark color, it is late wood. So we can say high density and is low density and lighter in color, it is early wood. High density and darker in color, it is late wood. So we know that on the basis of annual rings, we can determine the age of the plant. Now we will talk about hardwood and sapwood. It is hardwood and it is sapwood. Now there is hardwood and sapwood. And in case of hardwood, cell wall is hard. In case of hardwood, cell wall is hard. Because lignification of takes place. Lignin deposition takes place here. That is deposition of lignin takes place here. And this deposition of lignin makes this type of wood resistant. It makes the wood resistant. But in case of sapwood, there is no resistance because it is soft. It is having no resistance. Having no resistance from microbes because of its softness from microbes. And this also helps in, there is no xylem in case of hardwood. There is no xylem in case of hardwood. And this in case of sapwood helps us in conduction of water. It helps us in conduction of water. And there is no impact of microbial action. In case of hardwood, no impact of microbial action. And in case of sapwood, there is xylem present. There is xylem present, living tissues present, living tissue also present. And in case of sapwood, there is secondary xylem, peripheral, but it is secondary xylem is peripheral, and it is also lighter in color, but in case of hardwood, it provides protection and support. It provides protection and support. Protection and support. And there are no living tissue. There are no living tissues. Sorry, there living tissues are present. Living tissues absent. And living tissue present. No living tissue present. But in case of sapwood, there is living tissue present. In case of hardwood, there is no living tissue present, but in case of sapwood, there is living tissue present. Now there is another type of lateral meristem, which is responsible for secondary growth. It is called as core cambium. Another type of lateral meristem. Another type of lateral meristem, which is responsible for secondary growth. Responsible for secondary growth. It is called as core cambium. And this core cambium is also called phalogen. Now, when we talk of dicot stem, we have seen there are trichomes. For example, it is dicot stem, it is epidermis like this, and we know that in case of, when we talk of dicot stem, we know that there are trichomes. These are trichomes, and we know it is epidermis. And we have seen that this region is called as, I have told you this region is cortex region. It is cortex region. And then there is, when there are present trichomes. So in case of dicot stem, we have seen there are trichomes which are present upon the epidermis. So when there is cortex region, and this core cambium is present in the cortex region. Core cambium is present in the cortex region. For example, these are the cells of core cambium. It is present in the cortex region. Core cambium. And now there are the cells. This core cambium is made up of the cells. This core cambium is also called halogen. Halogen. Now there is, when this core cambium differentiates, core cambium have some specialized cells which differentiate. When outermost layer of stem damages, then sooner or later the cells need to repair. So there is a region, cortex region, and core cambium is present in the. Then there is a cortex region and core cambium is present in the cortex region. Core cambium have some specialized cells which differentiate. When outermost layer of the stem damages, then sooner or later the cells need to repair. This repairment occurs with the help of core cambium. When this core cambium differentiates and separates, then divide. When they differentiate towards outermost layer, it forms the structure which we call as cork. So cork is here formed. When core cambium differentiates towards outer side, cork is formed. And when this core cambium differentiates towards inner side, it forms secondary cortex. It forms secondary cortex. For example, these are the cells of secondary cortex. These cells of the secondary cortex are actually parenchyma cells. Parenchyma cells. Now we know that another name of secondary cortex is haloderm. It is haloderm. And core cambium is called a halogen. Now there is cork. Another name of cork is phellem. Phellem. So cork is called a phellem, secondary cortex is called a haloderm, and core cambium is called a halogen. So these collectively, these three are called a periderm. Collectively, these are called periderm. Now we know that cork is outer side, but a structure which is a technical term for which is given for dead cells. For example, I will show here the structure once again. These are the cells, and this is outermost layer. Like this, it is outermost layer. And due to the cambium activity, pressure radii becomes and helps to repair the damaged cells. At first, the Borg is formed. It is called as early Borg. When this cell differentiates, it forms pore-like structures here. These pore-like structures, small pores, which we call as lenticels. Lenticels. These are small pores present on the surface of stem. And what is the function of these lenticels? These allow exchange of gases. Allow exchange of gases. Exchange of gases between internal tissues and atmosphere. So we know that due to cambial activity, I have told you that there are core cambium cells. These differentiate towards the epidermis and separate, and then these cells form a structure which we call as Borg. So these are core cambium cells. So we know that these, due to the cambial activity, pressure radii becomes and helps this. Who helps the damaged cells? Pressure radii. Pressure radii helps the damaged cells. Helps the damaged cells.